To many, evolution seems to involve little more than a continuous change in shape and function from one life form to another. Early in the 20th century, D'Arcy Thompson superimposed a coordinate grid on a human skull. He showed that a simple curvilinear transformation would map the human skull to the skull of a chimpanzee. If we apply the inverse of the transformation to an adult chimp's profile, we find it changing into a form similar to a young chimp. This form looks somewhat human, but we are left with the question of how a youthful chimp might be transformed into a human being. One key human feature is the large brain, which might attain human size through the prolongation of infantile growth rates. However, the brains of intelligent people range over a factor of 2 in size, and the real question seems to be, how did the billions of neurons in the brain become organized to manifest unique human abilities? In the 1860s, Alfred Russel Wallace, the co-inventor of Darwin's theory of natural selection, noted that people of primitive societies have abilities as great as those of technologically advanced societies. He suggested that these abilities and the talents of unusual geniuses such as Mozart would not be favored by natural selection, and also could not be haphazard byproducts of the natural selection process. This led Wallace to supplement evolutionary mechanisms by divine intervention, a step which greatly shocked his friend Charles Darwin. Darwin declared that if it could be demonstrated that any complex organ existed, which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down. To illustrate his approach to explaining the origin of complex organs, Darwin proposed that the vertebrate eye began as a nerve connected to a light-sensitive spot. He then proposed a series of plausible intermediate eyes, stressing that extremely slight variations can take one continuously from the light-sensitive spot to the eye of an eagle. These rough sketches may seem superficially plausible, but one might wonder whether well-defined models of the eye could be constructed that would actually show Darwin's series of finely graded intermediates. Such models would have to show how neurons and photosensitive cells gradually became hardwired to perform sophisticated image processing and transmit the resulting data to the brain. The problem here is similar to the problem of brain evolution. We are dealing not with smoothly varying shapes, such as the human skull, but with discrete interacting subunits that fit together in a complex combination. Even the lens of the eye, far from being a simple smooth shape, contains complex layers of cells sutured together by zipper-like interlocking tabs. To gain greater insight into the Darwinian theory of evolution, we propose to study precisely defined mathematical models of the natural selection process. To do this, we first need to define complete working models of the biological systems we wish to study. To help elucidate the effects of mutations and natural selection, these models should demonstrate the operation of the biological system, and they should also demonstrate how the system forms or self-assembles under the direction of genetic coding. Once we have operation and self-assembly in our model, we can define the life cycle of the biological system. Natural selection is simply the change in gene frequencies that automatically occurs as the life cycle takes place, and some organisms leave more descendants than others. Under the heading of mutation, we lump together all processes which change genetic coding in a random way. The effect these changes have on biological structures will depend on the self-assembly process. We begin by introducing a simple model for the evolution of the eye lenses of a particular kind of trilobite, an ancient marine organism. Although trilobites have been extinct for millions of years, lenses from their eyes still exist, since they were constructed from the mineral calcite. A remarkable feature of these lenses is that they are corrected for the optical defect known as spherical aberration, By dividing the lens into two regions of slightly different refractive index, it is possible to cause light rays emanating from focus f1 to converge accurately to focus f2, thus allowing for a sharp image. This can be achieved if the interface between the two regions follows the ideal curve shown here. This curve was discovered in recent times by Descartes and Huygens, but it is closely approximated in the eyes of ancient trilobites. In our model, we will first assume that the interface curve can undergo a certain kind of random change as a result of genetic mutations. The curve to the right of a randomly chosen point is raised or lowered by a randomly chosen amount. We begin with a straight line as an interface and allow these random changes to occur. If the changes decrease the average divergence of the rays from an exact focus, then they are preserved. Otherwise, they are rejected. With this process of mutation and natural selection, we see that the interface does evolve to the ideal Cartesian curve. Now let us suppose that mutations affect the curve in a different way. Now, randomly placed short segments can be raised or lowered by randomly chosen amounts. Starting again with a straight line and the same selection rule, we find that the evolution process does not converge as quickly to the ideal Cartesian curve. Here we see the result of evolution by this mutation rule after 2,000 cycles of mutation and selection. For comparison, here is the result of the first mutation process after 2,000 cycles. The first type of mutation has its main effect on single segments of the interface curve, and thus it allows different segments to evolve more or less independently of one another. In contrast, the second type affects pairs of segments, and thus interface segments are forced to evolve in an interdependent way. This interdependence tends to slow down evolution, and it can lead to what students of nonlinear optimization call local trapping. As we shall see, trapping due to interdependence can have a profound effect on evolutionary processes. Thus, the self-assembly process, which was not explicitly considered in the model, has an important role to play in theories of evolution. To properly introduce self-assembly into our models, it is necessary to consider the cellular processes of heredity. Unfortunately, this is difficult to do, since the cell, which in Darwin's day seemed to be a simple bag of chemicals, is now known to be formidably complex. For the sake of developing a manageable and yet realistic model, we will therefore consider the evolution of the T4 bacteriophage, an entity that exhibits heredity and reproduction, but is much simpler than a full-fledged cell. The T4 phage consists of an icosahedral head and a tail segment to which is attached a base plate with tail fibers. The phage operates by attaching to the wall of a bacterium with its tail fibers, and then fastening the base plate to the bacterial wall. Once the base plate is attached, the tail contracts and drives a central tube into the bacterium. The phage DNA stored in the head then enters the bacterium through this tube. Once inside the bacterium, the phage DNA takes over the bacterial machinery of protein biosynthesis. Phage proteins are generated, and these automatically join together to produce new phages. This process involves the combination of protein subunits in a fixed order along fixed assembly pathways. The subunits making up the T4 phage are protein molecules, similar to this one, the enzyme carboxypeptidase. Such a protein consists of a long chain of amino acid molecules that maintains a specific folded configuration under the influence of molecular forces. In these diagrams, the amino acids are color-coded, and the sequence of amino acids in the chain is shown at the bottom of the pictures. Although the surfaces of proteins look random, they typically exhibit specifically patterned binding sites, which enable proteins to interact or join together in a controlled way. Here the sites involved in the joining together of alpha and beta hemoglobin are shown. The specific molecular patterns on these sites allow for the joining of these two molecules and no others. It is important to note that a binding site on a protein molecule may consist of widely separated parts of the underlying amino acid chain. Here this is shown for the active site of carboxypeptidase. It is therefore possible that two sites on a protein, shown here in green and blue, will be interconnected by several parts of the chain. Thus there is great interdependence among the parts of a protein, and changes in the structure of one part are likely to affect the whole. In making a model of a bacteriophage, we will abstract from real proteins the features needed to represent phage operation and assembly. A three-dimensional model can be made by representing proteins as ellipsoids with numerically coded binding sites on their surfaces. We can also consider two-dimensional models in which proteins are represented by rectangles with similar binding sites. An important feature of these model proteins is the process of conformational change. In this example, subunits A and B join together. A change is then induced in the upper bond site of B, which causes a further change to occur in its lower bond site. This change, in turn, allows B to recognize and join with C, which was previously not possible. Available evidence suggests that the rigidly specified order of assembly in the T4 phage is achieved by means of many conformational change linkages of this kind. Here is a three-dimensional bacteriophage model. Since the self-assembly of the T4 phage head is poorly understood, we have tried here only to simulate the tail and base plate of the T4 phage. The long DNA strand of the real T4 phage is replaced in the model by a short rod which runs from the base to the top of the phage. This DNA rod is surrounded by a coil of subunits which corresponds to the syringe-like tube of the real phage. The coil in turn is surrounded by a sheath of subunits which can be triggered to contract and drive the coil through a simulated bacterial cell wall. The self-assembly and operation of the phage are controlled by a network of conformational change linkages. As we can see from these two-dimensional schematics, this network is quite complex, with each protein subunit interacting with several of its neighbors. Here we see the self-assembly of the phage model, starting with a DNA rod and randomly positioned phage subunits. Subunits collide randomly with the growing phage and are added to it in an orderly way by the action of conformational change linkages. The operation of the phage model is also based on conformational change linkages, which trigger contraction of the sheath once the phage has attached itself to the simulated cell wall. The wall is modeled to simulate an elastic substance that breaks after being stretched beyond a certain limit. Both the phage and the wall satisfy a principle of free energy minimization, similar to that required in real systems by the second law of thermodynamics. What can we say about the evolution of a system such as this model bacteriophage? We can define a life cycle by allowing phages to self-assemble and break a variety of simulated cell walls. Phages that succeed can be selected for future life cycles, and random mutations can be introduced in the genes specifying phage subunits. What evolutionary transformations will arise from this process of natural selection and mutation? The answer to this question is that the strong interdependence of the phage subunits places severe restrictions on the ability of the phage to evolve. To see why this is so, we will analyze a simpler two-dimensional bacteriophage model. This model is also capable of self-assembly, and it operates by attacking a two-dimensional flexible barrier, breaking through it, and placing a small test molecule on the other side. This model is considered to have functioned successfully if the test molecule is transferred to the other side of the barrier and set free to wander randomly. The rectangular subunits of the two-dimensional phage are encoded by strings of symbols S, B, and K, where S stands for a neutral site, B stands for a bond site, and K stands for a corner. A string is required to fold to form a complete rectangle. A bond site consists of 18 bits and an optional sign, which allow bond sites to recognize and interact only with specific bond sites on other molecules. Any symbol in a bond site string is allowed to mutate with a probability of 10 to the minus 6th power per phage life cycle. Other allowed mutations include insertions and deletions of sites, which also occur with a probability of 10 to the minus 6th power per life cycle. This probability is based on known mutation rates in living cells. Here is a two-dimensional phage design that successfully breaks through a barrier that is 3 units thick. However, it is unable to break through a barrier that is 4 units thick. If we redesign the model by adding an additional subunit and making other related changes, we find that it is able to successfully handle both 3-unit and 4-unit barriers. In an environment containing both 3 and 4-unit barriers, a phage that can penetrate both will have a strong selective advantage over one that fails on 4-unit barriers. In this environment, will the 3-barrier phage on the left be able to evolve to a 4-barrier form, such as the one on the right? Analysis shows that to make the phage more powerful, it is necessary to add a new driving subunit. This can be done by duplicating the gene for one of the old subunits and modifying a minimum of 2 bond sites on the duplicates. This is the minimal change needed to add the new subunit, and this addition is only possible if the bond sites of the duplicated subunits are aligned one above the other. Without this prearranged alignment, additional mutational changes would be needed to add the new subunit. Taking into account the rate of mutations and the need for prearranged alignments, the highest possible probability for adding a new subunit is about 5 times 10 to the minus 19th power. If a new subunit is added, the resulting phage will fail to function unless the 2 side pieces of the phage are simultaneously elongated. This requires adding sites to the side pieces by site insertion mutations. The probabilities for these elongations come to about 10 to the minus 32nd power per side piece, given the assumed mutation rates. The transition from the 3-barrier to the working 4-barrier design requires that all of these mutational changes must occur simultaneously. The probability is therefore less than about 6 times 10 to the minus 84th power, a prohibitively small value. Actually, similar transitions in real complexes of interacting proteins are likely to be far less probable. For example, many simultaneous changes would be required to change the shape of a protein while preserving existing bond sites needed for interaction with other proteins. One might argue that exponential growth rates of microorganisms allow transitions of such low probability to occur. However, the Earth is not large enough for this. For example, the maximum number of E. coli bacteria that could live in a global ocean 8 kilometers deep is less than about 2 times 10 to the 34th power. Many other phage models can be studied. For example, consider this design
which operates by uprooting sections of the flexible barrier. Such designs can be readily created within the mind. However, it is difficult to see how they could originate or evolve significant new capacities in an environment where successful operation is required for survival.
In general, models involving many strongly interacting parts tend to require prohibitively improbable simultaneous mutations in order to evolve new functions or improved capacities. One might suggest that this limitation is an artifact of our model and not true of real systems, such as the T4 phage. If this is so, however, it should be possible to formulate phage models which are amenable to evolution, and this should be a topic for further research. Without explicit models of biological systems, the theory of evolution becomes simply an exercise in imaginative storytelling and not a proper scientific theory. We will end by briefly noting some examples of biological machinery to which this model-building approach could be applied. One example is provided by the E. coli bacterium. This bacterium is propelled by spiral flagella that are rotated by motors of molecular dimensions built into the bacterial cell wall. Can one make a working model showing how these motors might have evolved? To model the evolution of these bacterial motors, it is first necessary to understand how they work. Here is a model of the E. coli motor based on the work of Howard Berg. The model includes a wheel attached to an axle. There are several movable subunits around the wheel. Each subunit is mounted so that it can oscillate back and forth parallel to the wheel rim. Each subunit has an opening to the inside of the cell membrane, represented by the vertical tube. The subunit also has an opening underneath that faces outside the cell membrane, and a channel along the rim of the wheel that links the two openings. Because there is a difference in hydrogen ion concentration between the inside and outside of the cell, hydrogen ions tend to enter the subunit's channel more on one side than on the other. When they enter the channel, the hydrogen ions tend to bond to the wheel rim. The random oscillation of the subunit causes the sides of the subunit to strike the bonded hydrogen ions and give a push to the wheel. Since the ions tend to bind to the rim on one side of the channel, this side tends to hit the attached ions more often than the other side. As this happens with all the subunits, the net effect is to rotate the wheel in a particular direction. Our question is, how could a mechanism like this arise by mutations in a cell lacking such a mechanism? What would be the useful intermediate structures leading up to such a mechanism? Another example is provided by the cell walls of bacteria. These walls are made of sheets of cloth-like material. Each sheet is composed of a rectangular array of complex molecular subunits which are assembled outside the bacterium by the action of special enzymes. Can one make a working model of bacterial wall formation and show through what stages this complex system may have evolved? In the cellular reproduction of DNA, the DNA strands can become entangled. An enzyme called DNA gyrase, envisioned here as a molecular robot, is able to grasp two crossing DNA strands. By breaking one strand and reconnecting it on the other side of the other strand, this enzyme can tie or untie knots in DNA. Since this enzyme is essential for cellular reproduction, it would be interesting to make a model of its operation and its origin. Finally, we consider one of the many examples of higher organisms with features that seem to require many simultaneous steps in order to evolve. The statocyst or balance organ of this crustacean depends for its function on a small grain of sand inserted by the animal when it molts. For this arrangement to work, both the complex statocyst and the complex instinctive behavioral pattern seem to be required simultaneously. How did this come about? Our last example is the flatworm Microstomum that eats hydras and incorporates their sting cells into its own defenses. Here we see two hydras. In this enlarged view of a hydra's arm, we see sting cells and cells that fire sticky threads when triggered. Here we see an untriggered sting cell. The sting is coiled inside out within the cell. Here, the same cell has discharged its poisonous sting. The sting is ejected by pressure within the cell. The Microstomum can devour hydras, even though hydra stings can kill it if it carelessly brushes against a hydra's arm. The hydra tissue is digested by the Microstomum, except for the sting cells and the cells that fire sticky threads. These cells are swallowed by cells in the parenchyma, or stomach wall, of the Microstomum. The cells that fire sticky threads are then digested, but the sting cells are left unharmed. Each cell carrying a sting cell migrates to the back of the Microstomum. There the transporting cell is surrounded by an encapsulating cell. The sting cell is then oriented so that its sting is directed outside the Microstomum, and the epithelial tissue above the sting becomes very thin so that the sting can easily pass through. The sting cell is then ready to be used against the enemies of the Microstomum. The encapsulating cell is able to fire the sting cell when the Microstomum is agitated by an external threat. To benefit from the sting cells of the hydra, the Microstomum must correctly carry out each of several complex operations. Question: can you develop a model in which this defensive system evolves by natural selection and mutation? This lecture was first given in Budapest in 1987 at the International Symposium entitled Organizational Constraints on the Dynamics of Evolution. The models and analysis presented here are discussed in 3 books and a number of technical articles.